Moving on to type 3 hypersensitivity reactions. These are the product of antibody complex accumulation. We first identified type 3 hypersensitivity responses via the treating of diphtheria patients with horse serum, which is basically horses would have been infected with diphtheria, which would be a deadly disease. The horses are more expendable than humans, so some of them might die, but those that live will have, they would have had their innate immune response, and then their adaptive immune response would have kicked in, where they would have ended up producing plasma cells capable of producing antibodies specific to the diphtheria antigens. We would basically take the horse blood, we would separate the horse blood by taking out the plasma with the, we have a look here, we would take out the red blood cells, the leukocytes and the platelets to get plasma, and then we would take out the clotting factors to get serum. So the serum would mainly contain these immunoglobulin molecules that the horse produced to fend off the diphtheria. So these antibodies in the serum would be specific to diphtheria antigens. We would put the serum into humans. So the humans, if a human has diphtheria, the horse antibodies in the serum would target the diphtheria and opsonize it, and then our macrophages and neutrophils would recognize these antibodies and then destroy the diphtheria cells by phagocytosis, getting rid of the infection. The problem is, if we are exposed to the horse antigens more than once, we're going to have a type 3 hypersensitivity response. Because the first time that we were exposed to the horse antigen, our body recognized the horse antigen as a foreign protein, so we ended up with our own adaptive immune response to the horse antigen, and now we have B cells in our body that produce antibodies that are specific to the horse antigen that is specific to diphtheria. So if we ever come into contact with the horse antibody again, we recognize it as an antigen, and our memory B cells are going to differentiate into plasma cells that are going to produce antibodies specific to the horse antigen. So our antibodies are going to bind to the horse antibodies, creating antibody complexes which damage our tissues. So type 3 hypersensitivity reactions are caused by circulating immune complexes. Symptoms of the disease depend on where these immune complexes are deposited, and the immunoglobulins produced here tend to be immunoglobulin G and immunoglobulin M, the exact same as type 2 hypersensitivity responses. These antigen-antibody complexes form in a normal immune response, but get cleared by the liver and spleen. Excessive amounts of these complexes are formed in type 3 hypersensitivity reactions to either self-antigens, as in the case with lupus, where antibodies are being produced to nuclear antigens, so self-antigens from our nuclear contents that we're going to talk about more in a future video, or like we talked about with the type 3 hypersensitivity reaction to a serum with the horse antibodies specific to diphtheria. They form complexes and they're deposited in tissues. Deposition of these complexes in antigen-rich tissues results in activation of inflammatory cells like neutrophils. Basically, the antibodies bind to either the self-antigens or the circulating foreign proteins, like the case in the serums. They accumulate in the tissues, causing inflammation and tissue damage. Serum sickness is the scientific term for what we described with when our own body starts producing antibodies that are specific to antibodies from other animals that we've been in exposure to before. So we've been ill with diphtheria. We've had horse serum injected into us with horse antibodies for diphtheria that kill the diphtheria in our body, opsonizing it so our phagocytes can destroy them. But when we got exposed to these horse antibodies, our own immune system started to produce B cells that produced antibodies that were specific to the horse antibodies. So if we ever come into contact with these horse antibodies again, we have memory B cells that are going to produce antibodies when they come into contact with these horse antibodies, producing antibody-antibody complexes that cause serum sickness. And obviously you can imagine when these antibodies kind of accumulate, they cause tissue damage, inflammation, and that can be very bad in small areas with small blood vessels, like in capillaries or in the kidneys, in the glomerulus of nephrons. And if you look to the right here, I've just shown an image of a woman suffering from systemic lupus erythematosus with the characteristic malar rash. These have been quite dense topics, so I'd recommend taking note of the information on these slides. It's a little bit hard to explain without kind of having a whiteboard to draw these things on individually. I'm just kind of reading what can be read. It would be better if I had a whiteboard, but the notes will make more sense when you read over them on your own time. Now, finally, for the hypersensitivity reactions, we're going to talk about type 4, which is hapten-induced, which sounds complicated, but it's actually the easiest of the hypersensitivity reactions to remember, in my opinion. The majority of these reactions are caused by these molecules called haptens, molecules of small molecular weight that can penetrate the epidermis and bind to certain proteins in the skin. Hapten carrier protein complexes or antigen presenting cells will activate helper and cytotoxic T cells, as well as macrophages, resulting in inflammation, phagocytosis, and cytotoxic T cell apoptosis of antigen presenting cells. So examples of this include contact dermatitis. So you can see the image on the left is just this type 4 hypersensitivity response on a small scale. The little red circles are the irritants, so the haptens that are going to pass through the skin, the epidermis, get to the deeper layers of the skin in the dermis, where they are going to result in an immune response with helper T cells and macrophages releasing inflammatory cytokines and phagocytizing cells that are presenting these irritants. And then cytotoxic T cells are also going to destroy our own cells. You can see this on the right here with contact dermatitis. The haptens would have got onto this man's skin here, penetrated the outer layer of his skin, the epidermis, got to the underlayer of his skin, the dermis, where it's caused this swelling, which you can see like the red inflammation on his arm, as well as the destruction of some of his skin cells, which you can see by the flaky kind of dead skin cells on there. So helper T cells, macrophages, cytotoxic T cells have been killing his healthy skin cells because they've been exposed to these irritating haptens. Thank you.